Hello and welcome. I'm Claudia Rizzini, the Executive Director of the Radcliffe Fellowship Program at Harvard University. It is my pleasure to introduce our speaker today, Christina Warinner, our Starling Seaver Associate Professor. Christina deals with the archaeology of the invisible, also called biomolecular archaeology. This field of inquiry investigates decayed biological materials in archaeological and paleontological locations, along with ancient biomolecules, to gain deeper insights into our common human history. We probably never thought much about the remnants of sugars and starches that formed the plaque on our teeth. For Christina, the mundanity of plaque opens a window into the lives of prehistoric humans. Using dental calculus, commonly known as tartar, from ancient peoples, Christina unravels the mystery of human history and health through the entire world concealed in fossilized plaque. Christina's research findings have been instrumental in reconstructing the respiratory system and microbiomes of Neanderthal and Paleolithic peoples. Her insight chronicles the evolution of pathogens, demonstrating the intricate interplay between diet, infection, and immunity, and revealing the evolutionary vulnerabilities that can be used to better address contemporary human health issues. During her time at Radcliffe, Christina is working to understand the paradoxical relationship between humans and milk. We produce over 700 million tons of milk each year, enough that every single person on the planet could consume a quarter of a liter of it every day. Yet, most of the world's population is lactose intolerant. Christina's research is set in Mongolia, where she works with local herders to better understand the microbes present in dairy production processes. By identifying the microbes in the various processes herders use to create dairy products, she can use those microorganisms and clues to navigate the prehistoric microbial archive. Her work will help trace the origins of dairying and its migration across the world. The Q&A session today, the Q&A session of the public talk today, will be moderated by Ruth Grossman, our Mary Beth and Chris Gordon Fellow this year. And now please give a warm welcome to Christina Warinner.
Thank you so much, Claudia. It's a real pleasure to be here and to get to talk to you about some of my work. I've been interested in dairy and the history of dairy for a really long time. I've been working on it for more than a decade, and the story just gets more and more interesting, more and more complex, and more confusing. And so one of the things I want to do is try to bring a lot of the evidence, the information, the experiences that we have together in one place, because milk and dairy is a topic that's so interesting because it's studied in so many different domains, ranging from medicine to food and cuisine, nutrition, archaeology, anthropology, and many of these different groups don't actually talk together very much, and I think it prevents us from really seeing the big picture. So today I'm going to try and bring a lot of these threads together and tell a new story of the kind of history of dairying and where we are today with dairy foods, which are one of our oldest prehistoric inventions, but also one of our most fraught foods in the present day. Before I begin, though, I want to acknowledge that so many people have contributed to this work. So I lead a large team, or actually multiple teams, that have contributed in different ways to trying to tell this interdisciplinary story. And so I want to just show them right up front at the beginning of the talk that you're going to be seeing the contributions from them. They range from archaeologists, nutrition scientists, nomadic herders, ethnographers, historians of science. We've all tried to come together and kind of put our heads together and tell this bigger story. So one of the things that makes our species, or really our genus, different, so Homo, we're really different from a lot of other primates, from other mammals. One of the things that actually sets us apart from our closest primate relatives is we have tremendous dietary flexibility. We're omnivores, we're capable of eating many foods, and we do. We see this across populations all around the world where we have a huge diversity of cuisines that can be compositionally extremely different and yet all nutritious. And so we can survive on a really wide range of completely different dietary components. And this poses challenges and opportunities for us. And one of the more interesting foods, I think, that we created, it's a manufactured food, is milk and dairy products. As Claudia mentioned earlier, we produce truly an enormous amount of milk commercially every year. We produce nearly 700 million tons of milk each year. This is a really hard number or volume to wrap your head around, I think. So I put this into some more familiar units. So that's the equivalent of over 278,000 Olympic-sized swimming pools of commercial milk production every year. It is enough for every human on Earth to consume a quarter liter of milk every single day. And yet we know that most of the world's population actually has a lot of difficulty digesting fresh milk. So how do we get to the point where we have this food which is spread globally, it's eaten in so many different places. Today it's eaten on every continent in various contexts. It's a globalized food under huge production, and yet it's very difficult for us to digest. There's a kind of a paradox there. It's also confusing because we're mammals. One of the things that defines mammals is that our early lives are defined by consuming milk. So on the one hand, milk is the most natural and nutritious food a mammal can have, and yet the way we consume it is unlike any other mammal. So milk is very nutritious. It's really rich in a wide variety of nutrients. It's the only food for a young infant, whether that's humans or kittens or puppies. For a mammal, it's their sole source of nutrition for their early life, and it's rich in sugars and fats and proteins and a wide range of minerals and vitamins that are all designed to grow a growing infant. What a lot of people don't know, though, is the composition of milk is not uniform across mammals. It actually varies quite a lot from species to species, and I've just put a couple of examples here of different common species that are milked today, and you can see they have really different macronutrient profiles. So for example, humans have quite high amounts of sugar in our milk, relatively low amounts of protein. Horse milk is actually compositionally a little bit similar to humans, also high in sugar, which is why it's really good for making alcohol from, for example. Whereas other milks, like reindeer milk, is very, very enriched in fat and protein because that baby reindeer really needs to put on a lot of weight and grow very quickly in order to survive the upcoming winter. So they're really fine-tuned. The milks of different animals are fine-tuned to the needs of those offspring. And in addition, the milk composition does not stay the same over the lactation period. So it actually changes quite a lot over the development of the offspring, changing in composition over the period of months that the infant is being nursed until it is ready to be on other foods. So it changes throughout that course. But one thing that sets us apart from all other mammals is, of course, that we consume the milk of other animals. No other animal does this. This is something rather unique to humans. And also something that is unique to humans is we consume milk long into senescence, into old age. We don't stop in early childhood. So these are two things that really set us apart from other mammals. So what are the barriers here? Why don't other mammals just continue to drink milk? Well, there's a number of reasons, and the major reason that limits this is this disaccharide here, this sugar called lactose. So lactose is a disaccharide, so it's two sugars linked together by something called a beta-glycosidic bond, and that's just a really strong bond. It's very hard to break it down, and we need a special enzyme to do it. It's made of two sugar components, one glucose and one galactose. We have an enzyme that we produce in our small intestine. It's called lactase, and lactase acts as like a kind of molecular scissors that can cut that bond, and it's encoded by the lactase gene, which is on chromosome 2, and it's expressed in the small intestine, and it cleaves this bond, and that frees the two sugars to be absorbed by the small intestine. We actually can't absorb it in this disaccharide state. We have to cut it, so it kind of acts as these molecular scissors that cuts it apart so that we can digest it. And what's really important is the lactase gene is controlled, its expression is controlled by another region just upstream of it, actually in a different gene. There's a little regulatory region. It's actually quite small, a couple hundred bases. And this controls the turning on and the turning off of lactase. And this is genetically programmed, so young infants, it's turned on, and then it slowly turns off as we age. So what happens when you produce lactase and you consume milk? So if you're producing lactase, you consume the milk, it goes into your small intestine. Once it's there, your lactase cleaves it into simple sugars, you absorb it into your bloodstream, and it promotes growth, and this is how infants grow. However, if you don't produce lactase anymore, for example, when you're an adult, then what happens is the lactose will pass undigested into your large intestine, which is full of trillions of bacteria that are more than happy to help you digest that lactose. They can break that bond. The problem is, in the process, they'll produce about 8 liters of hydrogen gas for every quarter liter of milk that you consume. So this is part of what contributes to the symptoms of lactose intolerance. Okay, so let's go back in terms of the genetic underpinnings of this mechanism that we have for mammalian digestion of milk. So we have the lactase gene here, and then upstream we have this other gene, it's called MCM6, and there's this actually little intron region that controls the turning on and turning off of lactase. So when you're an infant, that is turned fully on. So that regulatory region turns on high lactase expression, and you produce a lot of it. But you're actually genetically programmed, and so are all mammals, to slowly decrease the amount of lactase expression as you get older until it becomes almost unmeasurable. It's never 0, but it can get very, very low. And this is part of the natural weaning process to help infants move off of milk and onto other foods as they age. But of course, we consume the milk of other animals into adulthood. So how is this working? How does this actually happen? There was a lot of interest in this, particularly in the 1990s and 2000s, to try to solve this question of how do we continue to produce lactase after infancy. And there were a number of studies that contributed to this. I'm going to kind of summarize more than decades worth of work. But essentially what they found was that they identified the region in the MCM6 intron that controlled this process. And looking at global populations around the world, they noticed there were these consistent variants, these genetic variants that appeared over and over again that were associated with milk tolerance. They are found in specific populations, and their distribution reflects their history and prehistory of the connections between these populations, and we'll talk more about that in a minute. So for example, this one here, if this C converts into a T, this can basically interrupts this switch so that the lactase is always expressed. It's just permanently on as if you were an infant your entire life. This is really common in European populations, also in Anatolian populations, and in northern South Asia, and also in Iran. There's another variant that you see here. This is associated with camel herders. It's really common in the Arabian Peninsula and parts of North Africa. There's another couple of variants that have been identified in Tibetan plateau populations. There's at least 3, probably more, that are found within East African pastoralist populations. And what's interesting is each one of these has actually evolved independently of the other. So this seems to be a region under a lot of selection in association with milk consumption. All right, and again, what it basically does, all of these mutations, although they're all different, they all have the same effect. They disrupt this switch so that you have the permanent infant state of high expression of lactase. All right. So this got really exciting when this was first discovered and really kind of worked out in the 2000s. And right around that same time, archaeology had a lot of ideas about how the current spread and distribution of lactose tolerance may have come about through the migration of ancient peoples during the Neolithic. Once we have the domestication of plants and animals and the spread of these populations, the idea was perhaps they carried that with them, and that's how we have the present-day distribution of these different mutations or alleles. And right around the same time, the first ancient DNA studies were starting to get powerful. And this is a study by a colleague of mine who's done a lot of work on this topic, Wolfgang Hack, looking at trying to recover DNA from these earliest Neolithic farmers and trying to see if they were responsible for the spread of these lactase persistence alleles, these variants. And there was a lot of work that really, it was such an exciting time in the 2000s. All this work was coming out. There was genetic studies linking the early European farmers to Near Eastern farmers. Reconstructing a population migration that introduced farming, that it wasn't just a kind of cultural spread, but it was an actually movement of people. There was also new studies that were showing that the domesticated cattle in Europe were actually related to the domesticated cattle in the Near East. So it seemed these people brought their animals with them. There was also also some really exciting studies looking at pottery, where they were able to recover milk fats from pottery, showing that milking and cheese making had taken place. And so all of this was starting to come together, indicating that something really fundamental was happening in the Neolithic that then led to the spread of lactase persistence across Europe, which was the first region really studied for this topic. And there was lots of modeling done with present-day populations and ancient DNA to try to estimate when this mutation first arose and at what time, and it was all linked to this very early Neolithic process. And a lot of the work here really was pointing towards this linear band ceramic, these early Neolithic farmers carrying this into Europe and spreading these lactase persistence alleles, and that giving them an advantage that led to them having a lot of population success and expansion over the continent and replacing most of the previous hunter-gatherers. So all this kind of research came together in 2013. Nature wrote a feature article called The Milk Revolution, written by Andrew Curry, who's a really excellent journalist. He kind of summarized all this kind of fever pitch of work that was so exciting. It was one of the first really stories that ancient DNA could tell, solve a real problem in the past. And so it was really, really exciting. And they made this nice infographic that kind of reconstructed the origins of dairying from the Near East and then spreading into Europe and kind of putting some times on this, all very, very early. And this fit really well within kind of models at the time of domestication and development of new culinary technologies. So done. We figured it out, right? We got the whole story. This is awesome. So this was all coming out when I was kind of finishing up graduate school, And so this was really exciting. We solved a problem. Around the same time, however, there were some major changes in the way that we do genetic sequencing that made our ability to work with ancient DNA much, much more powerful. And once we had this, we started looking at much larger populations, and we had much greater sensitivity to investigate this question really closely. And there was a problem, though. There always was a problem, even with the initial data, with this kind of idea. And the problem is there was actually one study that looked at these early Neolithic farmers in Europe, and they actually didn't find any of these lactase persistence alleles. But they said it's too small. It's like a really small sample size. We probably just missed it. Once we can do more individuals, we'll find it. It's fine. A few years later, with the new technology that came out, these 3 studies dropped like a bombshell on the field. They have hundreds of ancient genomes in it, all across Europe and Asia, and they conclusively showed there is no lactase persistence at all during the Neolithic. It does not exist. This was shocking, because what it meant was lactase persistence did not arise amongst the early dairy farmers, as had been thought. Europe's cheese-making linear band ceramic farmers were not lactase persistent, so they were making dairy products and consuming all this milk, but they actually didn't have any of the genetic mutations that would support it. Dairying preceded, it turns out, lactase persistence by more than 4,000 years. We have 4,000 years of dairying and no genetic adaptations at all. And so lactase persistence turns out to be completely irrelevant for the early history of dairying. So this opens up a huge question, because medically we explain lactose tolerance on the basis of these mutations, of these adaptations. And yet, for 4,000 years, people are dairying. They developed the whole technology. They developed this whole food purposefully, but they have no genetic basis to digest it. So how does this work? So this was really exciting to me, and an incredibly interesting question, and I wanted to dig into it. So if you look around the world at different groups of people who have long histories of dairying, you find them scattered across Eurasia and Africa, and many of these populations are associated today with lactase persistence alleles, but there's one that's not, and that's Mongolia. And Mongolia was always the example that no one wanted to talk about, because it was the example that never fit the model. And so I became really interested in trying to understand what was happening in Mongolia. Mongolia is a fascinating place. They have the traditional subsistence economy is nomadic pastoralism, nomadic dairy pastoralism. Between today, about 30% of calories come from milk and dairy products. In the past, even about a century ago, as high as 80%. They milk more species of livestock than any other country in the world. They milk 7 different species. And a lot of their economy is really centered around dairy production. So I started to wonder if there might be alternative ways of adapting to a dairy-based diet that isn't based on your own genome, but might be adapting through the use of microbes, whether that might be, for example, culinary microbes and through fermentation, or by adapting your own gut microbiome to be able to facilitate improved digestion. So I started a project called the Dairy Cultures Project. The idea was to combine ethnography to work with herders to better understand how they produce their dairy products, all the culinary practices around the foods that they eat, to do scientific studies on the gut microbiome, for example, also on the dairy products to understand what sorts of microbes were facilitating the fermentation or the breakdown of lactose in these products. And then also to bring in archaeology and try to understand the long history of dairying in this place. Because when we started, it really wasn't known how long dairying had been practiced. We actually asked a lot of people what they thought, and almost everybody told us since Genghis Khan. That's how long. But there's a lot of evidence archaeologically to suggest that dairy pastoralism is probably older, but there was very limited direct evidence for the dairy component. There was certainly pastoralism, but what about dairy? And so we brought in the tools of genomics and proteomics through archaeology to try to address this question. So we've completed 5 years of research. We have a lot of different outputs that we've done, and I'm really excited to kind of show you some of the major findings that we have as we try to put all this together in one big story. The first thing I want to address is one of our big questions is, when did dairying actually begin in Mongolia? Like, how old is it? And so what we did is we worked with archaeologists across Mongolia and the National University, and we put together a collection of individuals that we were going to sample through time and space. So we have pretty good coverage of almost the whole country and spanning 6,000 years of time. And the idea is we're going to investigate this and try to understand the history of dairying. And then this is just to give you a sense of the wide variety of archaeology. These are different time periods in Mongolia represented by some of the archaeological features. And so we started with genomics. We wanted to reconstruct the genomes of these ancient people with the first question of, do they have lactase persistence? I mean, they don't today, but maybe in the past they did. Could we look through time and see if they used to have it in the past, perhaps? And so we looked into this because if you look at present-day lactase persistence maps, blue here means 0 or close to 0. Mongolia is modeled to have no lactase persistence, so none of these genetic features that contribute to persistent lactase expression. When we managed to reconstruct 214 ancient genomes, and were able to genotype them, and we were able to look through time, and what we found was over the entire period of Mongolia's history that we could analyze, 6,000 years of time, they never had lactase persistence above a level of 5%. So it was always low, very, very low. At some points, it rises up to about 5%, and then it drops back down again. So it doesn't seem to be under selection. This is mind-blowing because within Europe, the common allele for lactase persistence in Europe is the locus in the genome under the highest selection in the entire genome. Of all the evolutionary pressures and forces that we have on our genome, that one has the highest one in Europeans. And here in Mongolia, where you have a dairy-based economy, it's not under any selection at all. And this is a real puzzle. What's also amazing to me is it's not because it wasn't there. We can tell, and don't worry about the text on this, it's just to kind of graphically illustrate. If you look through Mongolia's history, which we were able to do through the reconstructed genomes, it has a really dynamic population history. There's a lot of movement, there's migrations, there's new groups coming in, and these groups brought with them the European variant for lactase persistence. It gets introduced over and over again over this entire period, and yet is never selected for. So this is really interesting. It kind of breaks our understanding that we had about the role of these alleles in dairy adaptation. So let's go back. So we didn't answer our question of when did dairy in Mongolia begin, but we don't see any lactase persistence. Okay, is there another way we can do this? And so up until this point, what a lot of people had done if they wanted to identify dairy is they looked at pottery. As you might imagine, dairy decomposes very easily. There's not like a great archaeological record of dairy products like you would have for animal bone, for example. And so how do you find dairy from the past? So what people had done in the past is they primarily looked at pottery, and then they'd extracted milk fats from it. And it's quite difficult to do. It's a little bit indirect because all plants, all animals, just adipose fat has these fatty acids, but in ruminant dairy products, they have a slightly different isotopic ratio, so we can measure that. And you can detect dairy products that way. However, it's completely non-specific to species. You can't say which species it is, so it's a bit of a coarse method, but at least it's been used. You can identify dairy, and that was done in many places in the world. The problem is with the archaeology of Mongolia is that the sites that we are able to excavate, that we can find, almost never have ceramics. They're very uncommon, and so we don't have very much to test. But what is common is burials. So you see all these Bronze Age burial mounds here. They make the burials very conspicuous, and we're able to analyze instead the dental calculus, or the tooth charter on the surface of the teeth. And so this is something we started exploring, and my team developed this a number of years ago, methods for extracting proteins from dental calculus, which allows us to reconstruct the foods that people ate in the past, and in particular focusing on dairy. So we started looking for dairy proteins preserved in this tooth charter on the surface of teeth. It turns out milk is great for this because it has many proteins. It's very protein rich, and it has many different proteins. And of the ones that it has, this one here, beta-lactoglobulin, preserves extremely well. It's just sort of like a Goldilocks protein that has all the right biochemical characteristics for both preservation and detection using mass spectrometry. This is what it looks like. It has a lot of great beta sheets promoting preservation. This is a rollout of it, so you can see the sequence, and it has a lot of variants, sequence variants in it that actually allow us to distinguish different species. So we can distinguish a sheep milk from a horse milk from a cow milk and start to actually look at which animals were being utilized in the past. This is my lab. This is the mass spectrometer that we use to analyze this. Here are some of the people in the lab. So we run an ancient biomolecules lab where we have to use strict precautions to try to prevent any contamination from the present day as we kind of peel back these layers and try to recover these ancient proteins. This is what the data looks like when it comes off the machine. People often wonder, what does your data actually look like? But we can use that to then reconstruct the specific types of milk that were consumed from ancient peoples, and this is an example of one of the spectra here, and then we can use this and compare it to a phylogeny and do alignments and actually determine which animals were present, and this is kind of what one of our finished data sets looks like, and this is a real archaeological sample with all the different milk detected bits of milk protein that we had from this ancient individual from Mongolia. All right. So we can use this to try to identify when different dairy animals, not only when did milk become important, but when the different dairy animals become part of this economy. So we went back to our data set and we applied this. And what we were able to find is we start seeing milk at 3,000. This is really interesting because this is when we have the appearance of the Afanazevo. And the Afanazevo are a group of people that migrated from the west, from the North Caucasus region between the Caspian Sea and the Black Sea. They settled in this region. They weren't particularly successful. They didn't last very long, but they seem to have introduced dairy pastoralism as a technology to Mongolia, which is then widely adopted by the rest of the population. Later, we start seeing horse milk, and then in the medieval period, we see camel milk, and a colleague of mine recently identified yak milk around the same time. So we see that this kind of dairy culture is coming together in stages. And this allows us to kind of reconstruct or reimagine the kinds of foods and foodways that were present at different periods in Mongolia's history and prehistory. All right, but how did it begin in Mongolia? I mentioned that it came from this region in the North Caucasus. We know that dairying actually begins in the Near East. So it begins somewhere around Anatolia, Levant region, around 9,000 years ago. Pretty early, people move it to Europe, although they're not lactase persistent. It also moves pretty early to Africa. And it also moves into the South Caucasus and then moves across into the North Caucasus. So by 6,000 years ago, we have dairying practiced in this North Caucasus region. It also goes to the Iranian Plateau. We don't have great dates, though, for that. What's really interesting about this region, however, this region, which is small, turns out to be massively influential in the Bronze Age. They develop the first carts and wagons. They develop a form of fully mobile pastoralism. And around 3000 BC, they become hypermobile. Groups from this region go west, where they completely transform the genetics of Europe and actually replace between 40 and 80% of the genetics of Europe. from this region. You also have groups that go east and form the Afanazevo groups, which introduced dairy pastoralism into Mongolia. So this group has a massive influence on basically laying the foundations of today's globalized dairy culture. It comes out of really this region. It was very small in the Neolithic and probably very ephemeral. It's out of this Bronze Age massive expansion that we get the foundations of the present day globalized dairy production. And we see it in Mongolia by about 5,000 years ago. We investigated this region more closely. We can actually really time it now, so we've been able to show exactly when it comes in. And what's really interesting is that the earliest dairying seems to be entirely focused on sheep, and then a little after 3,000, it diversifies, and you start seeing more animals brought in, and that's kind of, I think, important. We can talk later about sheep milk and why it's special. And again, this is really interesting because we have lots of movement of groups from this region repeatedly over the next several millennia that keep introducing new dairy products, but also these alleles. This region is also likely where the lactase persistence variant that is found in Europe today actually originated from. All right, but why dairying? Like, why do people want to do this? Why would you go to so much effort? And like, why specifically these regions, right? So today you can get all sorts of foods. You can go to the grocery store and you can eat food and you can get whatever you want. But at this time, people had to adapt to their local environments, and you had to produce foods that you were capable of making in these environments. The problem is this whole Eurasian steppe zone that I've shown you is a really challenging environment for humans to live. It makes great grass, and we cannot eat grass. You can't grow crops there, or very few, because the growing season is so short. In Mongolia, the frost-free season is only about 3, maybe 4 months. There are very few crops you can grow that quickly, so you're really limited. It's this huge expanse of land, but it's very hard to live on. But what's amazing is that if you can control ruminant animals or equid animals, they are so adapted to eating grass, and then you can eat their products. So then you can move into these huge expansions of space that previously were barely occupied because they could only support sparse groups of hunter-gatherers living along riverine corridors because these huge grasslands were essentially food deserts for them. But if you can control ruminants or equids who can convert the grass into nutritious milk, suddenly you can expand into all these environments. This is likely what's driving this huge expansion of dairy products. And what's really interesting is while in Eurasia, this adaptation to steppe and grassland is probably what's driving this, in other parts of the world, it's also really great for adapting to mountain environments and also fantastic for deserts. All right, so let's go back. So this question we had at the beginning, could microbes be part of the key to understanding how an alternative way of adapting to dairy products? So we first looked at the dairy products themselves, we characterized them, we measured the lactose components, we've been isolating bacteria from them and trying to understand how they worked. And while fermentation, which is like microbial consumption of the nutrients in milk, they do break down some of the lactose, it's not that much. And in fact, some of the groups we worked with at the height of the horse milking season, for example, in Bulgan province, some of the men we documented consume 250 grams of lactose every day. Huge amounts of lactose are being consumed. So clearly, the fermentation that's happening while it's reducing the lactose is not sufficient to explain the tolerance. All right, so we started working more with herders and trying to understand the different ways they're practicing dairy, and we also worked in multiple regions because we wanted to get an understanding of regional effects since they do milk so many different species. We also did a gut microbiome study to try to understand how the gut microbiome might be adapting. So this is a photograph I forgot to mention. This is one of my favorites. This is Gerald. She's showing us this. This is a still. She's actually making yak yogurt vodka. This is a 100-year-old still that's been used in her family. So you can really make an enormous variety of dairy products of all kinds that you've probably never tasted. In fact, most of the things that are consumed in Mongolia have no English translations. We also did a comparative study with alpine traditional dairy production, comparing herd management, also cooking and culinary practices to see what similarities were there and what differences were there. And we ended up mapping out a lot of the different production techniques in a kind of chain repertoire of how dairy products are made in different Mongolian households, depending on which animal and the specific goals of production are. All right. So for our gut microbiome study, I partnered with my fantastic colleague, Sonia Kishin-Solomon, who's a nutrition scientist in Mongolia. And we had 3 field sites that we were working in, one in the far north, which are cattle and yak herders, in kind of more centrally in Bulgan, which are horse herders, and in the south, which are mainly camel and goat herders. And we also looked at the capital city, Ulaanbaatar, which is a urban skyscraper city. And interestingly, almost all of the dairy produced there is commercially produced, and they import all their microbes from Europe. So they don't actually use any local microbes in the fermentation. It's entirely imported from Europe. We did a study where we collected gut microbiome samples, but also did comprehensive nutritional sampling and testing. We also did something called a lactose challenge test. This is a clinical test used to determine lactose intolerance. And on the basis of the genetic profile, theoretically, everyone here should test positive for lactose intolerance. And so we administered this test. You simply have like a night of fasting, and you consume a small cup that has 25 grams of lactose in it. And then we measure the amount of hydrogen that you breathe out over the next 2 hours. And that allows us to gauge the degree of colonic fermentation. Because if you are digesting it earlier, then you will have very little lactose will reach your colon. So you should have almost no hydrogen production, or if it's going undigested and being digested primarily in the colon, then you should see a large amount of hydrogen being produced, like we talked about earlier. And so we did this, and we had some really interesting results. So first of all, not everyone had high hydrogen. So about a third of the individuals actually didn't produce hydrogen at all. And if you broke that down, it was really different between the capital city and the herders. So the herders actually produced, many herders had very low hydrogen expression. So they weren't having any symptoms at all or any like clinically measurable effects of lactose. We ended up doing really deep sequencing. We sequenced over 17 billion sequences of the gut microbiome samples from these individuals. We were then able to reconstruct more than 9,000 microbial genomes, which now we're mining and trying to understand if we can link any particular genes to any particular functions that we're seeing that might be adaptive for milk digestion. And in the process, we've also identified many new species that haven't been described before that, at least at present, seem unique to these Mongolian populations, and we're really excited to understand how they may be contributing to lactose tolerance. One of the things that we do see is that there is a distinct gut microbiome composition. So the composition of the microbes in the herders versus individuals in the city are distinct from one another. They have different ecological composition. And they also are, if we look at kind of differential abundance, so which microbes are really enriched in some populations versus others, we see that the herders are really enriched in a few groups of bacteria that are pretty interesting. They're really enriched in lactic acid bacteria that are known to be present in the dairy products. So these are a lot of probiotics, and they're highly enriched in their gut microbiome, and they may be facilitating some of this digestion. They're also really enriched in a group of bacteria called bifidobacteria, which are really dairy specialists and are especially abundant in young infants. And one of the interesting functions of bifidobacteria is that when they metabolize lactose, they do not produce any hydrogen. We also found some really interesting patterns that were more regional specific. So we were really glad that we didn't just do one location, because we actually saw a lot of variability across the different regions, which may be related to the animals or the different climatic environments. So for example, Bifidobacterium ungulatum, which is one of these bifidobacteria that I mentioned, was particularly enriched in Hovsgol and in Dungovi. So this is in the far north, the yak herders, and also in the camel herders. But we didn't see it in the horse milk drinkers. Conversely, we saw that Lactobacillus helveticus, which is actually also found in Swiss cheese, hence the name, was really high in the horse milk drinkers, but not in the others. And then we found Lactobacillus strelbrueckii, which is a really common iconic microbe found in yogurts, was found to be very high in the northern populations, the yak and cattle herders, and also in the horse herders. So we're now trying to understand these microbes, and we've noticed that they actually have a lot more genetic diversity than we see in commercial strains. We're trying to understand how, even though they're familiar microbes, how they might be different or functioning different in the gut microbiomes of these individuals. One of the things we also want to do is we're trying really hard to establish a culture collection in Mongolia. One of the problems they have is they don't have a microbial culture collection, which makes it very hard for Mongolian microbiologists to do any of this study themselves in their own country. So this is something we are trying really hard to build up. Also, as I mentioned before, one of the problems that we see is that the dairy products that are under commercial production for the city people, they import all their microbes. this kind of has two problems. For one, they have plenty of amazing dairy microbes in their own country that they're not utilizing, and they're paying other people to license other strains. But it's also introducing these microbes into this otherwise quite fragile microbial ecosystem that's maintained by the herders, because sometimes what herders do if their microbes fail is they take a commercial product and they use it to reseed their starter cultures. This can have huge implications of wiping out these heirloom microbes which have been maintained in Mongolia for millennia. And so one of the things we're now trying to do, we have a project now where we are working with herders and we are isolating these bacteria, culturing them with the goal of creating a nonprofit so that we can then license these to the commercial producers, hopefully, and the herders can benefit from their traditional knowledge. This is something that's under development and that I really hope we can get off the ground. All right. So I hope I've given you kind of a sense of how interesting milk and dairy products are and how there's more than one way that humans have adapted to consuming milk and dairy products. It's not simple, and there's many different ways that we can adapt to different foods, and this is characteristic of our species being very flexible in the foods that we're able to eat. So there's one trajectory, which is to alter the human genome, and we've seen this in a number of populations around the world. But an alternative pathway seems to alter the microbes that we interact with, both through food and through our own gut microbiome. And really what's interesting about this is this seems to be the original way by which humans first adapted to dairy products and developed the dairy products in the first place. This leaves open a question: is what changed to then cause the genetic changes that occur in some populations? Those genetic changes occur rather late, and we don't know what the trigger was for it, or why in Mongolia, even when it's introduced, it's never selected for. Those are open questions that we hope to resolve. And so I hope I've given you a window into how also archaeology can help inform present-day issues or problems or challenges. And if anyone is interested in learning more about our work, we have, you can use the QR code here and visit our website where we have lots of related materials, including coloring books for children that explain how we do our work. We have a soundtrack that we've made of Mongolian folk songs about dairy products. We have videos about our lab. We have photo books and a number of short documentaries about the lives of herders and the way that they make dairy products. So thank you so much for your attention. And I just want to acknowledge the different funding sources we've had, and especially my RRP fellows who have been so critical to making this work a success. So thank you so much.
Thank you very much, Tina. That was fascinating. I have reams and reams of notes. And those 8 liters of hydrogen produced for every quarter liter of milk has given me a whole new set of compassion for my lactose intolerance friends. So thank you for that. Thank you also for the questions that have already come in. Please continue to submit your questions via the QR code in the room or online. So first question for you. Is there any variation, just given the 7 species that they milk in Mongolia, which is fascinating in and of itself, but is there any variation in the digestibility of milk from different species?
Oh, that's a really good question. Yes. Yeah, there is. So there's a couple ways of addressing this. So how do I start here? So one thing that's actually quite important, I didn't have time to talk about it, but I think it's fascinating, is the fat content of the milk has a huge impact on how you digest it. And the reason for that is it really slows down gut transit time, so the milk moves much more slowly through your body. That has 2 effects. That allows that residual amount of lactase that's still there longer time to act, but it also means that the lactose that is introduced into the large intestine is introduced at a much slower rate. So even if you do have lactose intolerant symptoms, it kind of ramps up more slowly than if you were to drink skim milk, which means that it moves almost at the speed of water and you get a huge lactose load all at once. So some of the milks are much higher in fat than others. So sheep milk is quite high in fat. And as you saw from the work that we did, we saw that the earliest dairy farmers that were really intensively dairying were focusing on sheep. And I think that's not a mistake. I think that sheep milk is more digestible for that reason because it slows the digestion. But yeah, so the lower the fat content, the more trouble people will have. And then you can also use them for very different things. So like horse milk for example, you can't make cheese out of it. You can't actually get the proteins to curdle or to form a curd. So horse milk is almost exclusively consumed as a liquid product and usually as an alcohol because it has very high lactose content and it makes a nice alcohol. And because it doesn't curdle, it doesn't separate. So it makes a really nice alcohol. And so other dairy products are used for other purposes. And then I didn't mention this either, but in addition to lactose sensitivity, some people have protein sensitivity to milk. So that beta-lactoglobulin that I showed you is actually quite allergenic. Quite a lot of people actually have an intolerance to beta-lactoglobulin, and that's because we don't make it. We actually pseudogenize that. So we have a series of mutations in our beta-lactoglobulin gene, which renders it non-functional, and so we don't make it. So our milk is free of beta-lactoglobulin, but the animal milks have it. So some people develop an allergy specifically to that protein. Interestingly, camels also lost the ability to produce that gene. So people who are sensitive to or have a milk allergy can drink camel milk usually.
All right, thank you. I'm still stuck on the yak yogurt vodka.
I meant to bring it. I have a bottle. I was going to bring it in for people to taste. It's actually really good. I think it's really nice.
I think I'm just fascinated by the thought process that led to the creation of yak yogurt vodka. But anyway, do you think actually that some populations in the past would consume dairy despite intolerances? And could that have possibly led to some of the changes in the microbiome?
Yeah, I mean, I think it is a really nutrient-rich food. I think you have to balance the consequences of it. I think if you're in a really precarious state already, then having that additional diarrhea and pain is probably going to be very difficult for you. But if you're a well-nourished population, you can probably handle it. One of the other things, too, that is important here is in Mongolia, they have such a dairy-based diet that there's very little fiber in the diet, very, very, very low amounts. And so constipation is a huge problem. So I think having a little bit of diarrhea actually kind of counteracts that a bit and may kind of neutralize those effects so that they're not as extreme. I mean, one of the things that was so interesting to me when I first went to Mongolia and was interviewing and talking with herders is they had absolutely no idea what I was talking about. Most people we talked to had never heard of lactose intolerance. This was not on their radar as a thing that existed. And they were very puzzled by it. And they were very excited to show the world that they don't have it and to participate in the study. And they were shocked to learn that that there's these different maps that have been produced by different health agencies that indicate that they're lactose intolerant because they don't have any concept of it. It's not something that is even a salient medical condition. It is becoming so in the city where it is on the rise, but I think that might be due to dietary changes and losing that adaptation to a dairy-based diet through microbiome changes.
Okay, thank you. Can you actually move your microphone up a little bit? We're getting some clicking with your hearing. All right, thank you. So do you know whether the differences in the herder's gut microbiome actually facilitate milk digestion, or is it a result of drinking milk, or how does all of that work?
That is such an excellent question, and I wish I could answer it, but I can't. I actually have one of my colleagues in the audience here today, Rachel Carmody. This is something we hope to be able to dig into. Right now, we can't tease it out. We don't know if it's a cause or an effect, but we're hoping to do further studies to narrow that down.
Okay. Do you think it would be possible to reverse lactose intolerance in adults using some of the non-hydrogen producing gut bacteria that you find in Mongolia?
Yeah, they do that already in Mongolia. This is something that I was also fascinated to learn about. There's something in Mongolia called a milk cure. So it does happen that people will develop an intolerance to milk. They don't think of it as lactose intolerance in the same way we do, but people will say they're having gastrointestinal problems, they're not digesting things as well, and they'll go on something called a milk cure. It's kind of like a spa, and you go and you consume horse milk, which is considered the best milk for this, and you consume it for about 10 to 14 days. And at the end, you restore your digestive ability. So this is something that's traditionally practiced in Mongolia. And I'm told that the initial stages produce what sound a lot like lactose intolerant symptoms, but over the course of this treatment, they subside and then go away. And so this is a long-standing traditional medicine in Mongolia.
Thank you. What are some theories of early dairying in Europe when this genetic polymorphism was not present? And is the presence of lactase-producing microbes in the gut biome there? Is there any evidence supporting that? How did that get started?
That's a great question. We do not know. It's kind of $1 million question what's happening in Europe, because it doesn't make as much sense in Europe as it does in other places. So, for example, the allele that's really common in Bedouin populations, especially in the Arabian Peninsula, like we can explain that because it's highly associated with camel milking, and so you can understand how that might be selected for. Because one thing that's quite interesting about camels is they can consume saltier water than humans can. So if you have a lactating camel, you can actually go further into the desert, access more oases that would be too brackish for humans. You can have longer caravans, you can have more territory. And so there's an understandable relationship between this selection pressure on that particular allele for being able to consume fresh camel milk. Also in East Africa, where milk is also consumed largely in liquid form, it's a very arid environment, and so they're utilizing the milk primarily as a source of clean water. And so you can see how that would be selected for. Mongolia, it's kind of different, but Europe doesn't have any water shortages. There's not really, it's not clear what the pressure would be or why suddenly you get the lactase persistence selected for rather late. It appears in the late Bronze Age and doesn't really undergo strong selection until the Iron Age. I don't know why that would be. I suspect it may have a lot to do with technologies that people are developing around cuisine. Because it depends, if you can prepare the foods in ways where you strip out the lactose. So butter is almost lactose-free. Lactose-free product. Hard cheeses are lactose-free largely. But it does appear that a lot of the iconic European cheeses that we think of probably are a very recent invention. They probably get invented in the Middle Ages. They likely don't have a long history. And so we do think during these very early phases in Europe, they probably are consuming soft cheeses or yogurts or something of that nature, which would still have lactose in it. So we don't know. It's really quite a bit of a mystery right now.
Interesting. And sort of tangentially, I guess, following up on that, is there any work that talks about the diversity of milk products that the Mongolians consume, which you've talked about a couple of times already, as being actually more helpful for the evolution of this microbiome?
Oh, that's a good question. I don't know. I think that's a fascinating question. I mean, one of the things we're trying to do is just track the movement of these microbes because one of my all-time favorite microbes, if one can have one of those, is a bacteria called Streptococcus thermophilus. I love this microbe. It's part of what defines yogurt. So in the United States, if you want to put yogurt on a label, it has to have Streptococcus thermophilus and it has to have Lactobacillus delbrueckii. Those are the two microbes it has to have to do that. And they are so interesting because they do not exist together in nature. Lactobacillus delbrueckii is a plant microbe. It's probably picked up by the calf or like the juvenile animal is probably getting it in its mouth by eating plants and then suckling, and then it's introducing it into the milk. But Streptococcus thermophilus is a salivary bacteria. It's a mammalian salivary bacteria. And the closest species that's ever been found to it is actually in our saliva, although I don't think that it's directly related. It's probably related to the juvenile offspring saliva. And so I think that's really interesting. I mean, one thing I would love to do is, theoretically, they should be different enough, the different strains of Streptococcus thermophilus should be different enough amongst the different dairy species that you might even be able to trace back the origins of some of these microbes that have now persisted in these traditional nomadic pastoralist dairy settings. This is something I'm really, really interested in doing. Can we actually, kind of excavate back some of that history through microbiology? Mongolia is also a very special place to do this because it is one of the few places in the world where they have a relatively intact traditional dairy production system. In many other places in the world, in the mid-20th centuries, there were really massive efforts to sanitize, sterilize, and replace these sort of heirloom microbes with high-performing commercial varieties. And so we've lost a lot of that genetic diversity, that microbial diversity in most places of the world for dairy products, and there's only a few places where you still do have really long-term maintenance of these microbes. And it's really interesting, these microbes that are under cultivation in Mongolia, they're extremely robust. They They can cultivate them every day over the whole course of the season. They freeze them over the winter and revive them in the spring, and they do this over and over again. If you were to take commercial starter cultures that you would order from a company, you can usually only get that going for a few days before it fails. They're high producers, but they're very weak and very they don't last very long. You have to constantly buy new ones, these lab-grown strains. Whereas in Mongolia, they have these very, very robust communities, and they're barely studied. And so that's something we're also trying to raise awareness for in terms of this form of cultural heritage and to study these microbes because they are under threat from climate change as the lives of herders become more and more difficult every year.
Okay. And actually, there is a question about climate change in here, so we can talk about that. Just what is the impact of climate change on the work that you're doing on these cultures in Mongolia and the persistence of dairying?
Their lives are changing a lot. I mean, the kind of dairy pastoralism that's practiced is extremely sustainable, and it requires very few inputs. The herders are very self-sustainable, but climate change is a huge threat for them. The major problem they're having there is the winters are getting much colder. On a normal year, it's minus 40 degrees Celsius in the winter in Mongolia. It's getting worse. And it's getting drier and more variable. So sometimes they have way too much snow, sometimes they have none. This past year was particularly bad. They had, I think, almost 2 meters of snowfall, and that's catastrophic for these herders because the animals can't get to the grass. It's so deep that they starve. And so this year, actually, one of the herders in our study, who was on the second slide that I showed, Loeb, who has an amazing camel herd. He's a very successful, very respected herder in Dungovi, and he lost a lot of his camel herd to the intense snowfall this year. And it used to be that these events would happen every couple of decades, and now they're happening every few years now. And this is really driving a lot of herders. They lose their herds. They don't have, their wealth is in their herds. They don't necessarily have bank accounts. Like their wealth is their animals. And if they lose the animals, it bankrupts them. And then this drives a movement to the city where a lot of them are impoverished, and there's not really an infrastructure for that. So it's a huge problem.
Okay. Can you talk a little bit about any relationship between this dairy consumption and life expectancy, bone density, healthy lifespans in general in Mongolia, and also probably, more broadly?
Oh, that's an interesting question. I think it's hard to talk about lifespan longevity because there's so many other factors that are at play. So herders don't have excellent access to healthcare. So it's very difficult to compare across other societies that do. So I'm not sure I can completely adequately address that.
Okay. So just looking at the time, what is the role of pasteurization in human microbiome evolution and lactose intolerance?
Oh yeah, so pasteurization is really interesting. Pasteurization gets invented because of the difficulty of supplying urban environments with country milk in the 19th and early 20th century. There's an amazing and kind of horrifying history of trying to provision cities with milk in the US and other places where the milk was quite contaminated and adulterated and was really dangerous. And so pasteurization was invented to completely sterilize the milk through heat and pressure so that you could stabilize it. And they also homogenize it too, which means you shake up the fat globules so that it doesn't separate as easily. And it makes it more stable, and so it could be transported over a longer distance. Pasteurization, though, is not particularly conducive to cheese making, and it makes the whole system a lot more difficult. They don't pasteurize in Mongolia. They do heat the milk. They do heat it, which does kill some of the pathogens, but a lot of the hygiene that they're able to achieve is actually through the extreme overgrowth of the lactic acid bacteria. So lactic acid bacteria are a group of bacteria that are highly tolerant of acid, and they produce a lot of acid. Most bacteria are not. Most bacteria are actually killed very easily with acid, which is why lemon juice is such an effective cleaner. And in fact, most of the bacterial pathogens and viral pathogens are very easily killed, even in mild acids. So by promoting the growth of these lactic acid bacteria, which are all the kind of flavorful culinary bacteria, they're able to maintain quite nice hygiene standards through a combination of heating, not pasteurization, but heating, and then through the maintenance of these microbes.
Okay. And I think just for one more question here, what do you consider to be the biggest mystery or biggest mysteries about milk that still exist?
Oh gosh, there's so many. I still do not understand how milk is digested. I mean, just on a basic level, there are so many factors at play. I mean, I really want to understand gut transit time better. I really want to understand if microbes, if some of these culinary microbes are surviving the stomach, because if the lactic acid bacteria they're acid tolerant. If they're surviving, if they're actually active in the small intestine and contributing to digestion in that way, that's something we don't actually know. I really want to understand, what actually is a probiotic doing, and can we even define that better? Probiotic is an amazing term with a very loose definition, and I'd like to dig into that more. And I really also want to understand why we suddenly get selection on the lactase persistence alleles in Europe, for example, in the late Bronze, early Iron Age, when it hadn't really been under selection before that. Why then? That's a big question for me.
So many things still to dig into.
Yeah.
Thank you so much for this wonderful presentation and all the interesting facts you've shared, as I'm going to have to go through my notes and ask you more questions afterwards. I really want to thank the audience as well for all of your wonderful questions, and I hope you'll be able to join us for other Radcliffe virtual programs. You can find out more about future programs at radcliffe.harvard.edu. Thank you very much, and have a wonderful rest of the day.